Welcome. In this presentation, I will tell you the differences between endotoxin versus toxic shock syndrome toxin. What is the superantigen mediated sepsis? Toxic shock syndrome toxin 1 is produced by the Staphylococcus aureus. This is a powerful superantigen that drives massive T cell activation and cytokine release. Interestingly, Streptococci pyogenes also produces a group of exotoxins called streptococcal pyrogenic exotoxins. They both act in a similar way, functioning as a superantigens and causing sepsis. Streptococcal exotoxins are not identical to toxic shock syndrome toxin. Because of this difference, the streptococcal toxin are referred to as toxic shock syndrome like toxins. How do they do this? Normally, when the antigen presenting cell, when the MHC complex, this is a peptide binding site, the MHC complex will express the peptide and the T cell receptor will get activated. Consequently, with the primary stimulation of the T cell receptor and the MHC complex, the T cell activation will happen. In superantigen mediated sepsis, the superantigen will bridge the MHC class 2 on the antigen presenting cells to T cell receptors. By doing so, it bypasses the normal antigen specificity, causing polyclonal T cell activation, resulting in massive cytokine storm, leading to the typical septic shock, fever, hypotension, and characteristic rash. The take-home points: both Streptococcus aureus and Staphylococcus aureus and Streptococci pyogenes produces the toxins. They are the exotoxins, and here we have seen about the toxic shock syndrome mechanism. This is the exotoxin which we had talked now. There is another entity, that is the endotoxins can also produce septic shock. How? Endotoxins are constitutive elements of the bacterial membrane. They are only liberated when the bacteria die. In the bacterial membrane, that is in the endotoxin, the lipopolysaccharide is the bacterial membrane. It has a component that is the lipid A. The lipid A component of the lipopolysaccharide will bind to the CD14 receptor, which is a component of the toll-like receptor 4. So this is the toll-like receptor 4 and this is the macrophages with the toll-like receptors. So when the endotoxin binds to the toll-like receptors, to be more clear, the lipopolysaccharide of the endotoxins, which subunit of lipopolysaccharide? The lipid A subunit of the lipopolysaccharide of the endotoxins will bind with the lipid CD14 receptor, which is a component of the toll-like receptor 4 signaling. Once this is activated, there is massive cytokine release, causing systemic inflammatory response. Here the T cells are not involved yet. With only macrophage activation, the endotoxins will result in a shock-like syndrome presentation. Both the shock will have high fever from interleukin 1 and tumor necrosis factor, hypotension, multi-organ dysfunction, capillary leak. But the bacterial sources are different. The rash is more common in toxic shock syndrome toxin. There is a lab pattern. In the exotoxin, there is a neutrophil predominance, but the lymphocytic predominance happens in the endotoxin. But this is not the rule. This is just you can see this pattern, and there is a different response to different therapies. Endotoxin shock is caused by gram-negative bacteria like E. coli, Pseudomonas, Klebsiella. Superantigen shock is caused by Staph aureus, Streptococci pyogenes. Here the primary immune cell is macrophage monocytes, but over here it is T cells and antigen presenting cells. This will involve only the macrophages, that is the CD14 and toll-like receptor of the macrophages, while the superantigen shock will involve the MHC2 and T cell receptors. Cytokine profile is almost the same. Endotoxin shock will cause an indirect effect, but superantigen shock will as such cause a massive polyclonal activation. Both can result in almost similar clinical presentation with the fever, hypotension and shock. This presentation is in short about the differences between endotoxin as well as the toxic shock syndrome toxin. I have tried to make it as clear as possible. If you like my presentation, let me know by giving a thumbs up. All the best for your exams.